The overall goal of this procedure is to identify functional Qa-1 epitopes in proteins.
This method can help answer key questions in the field of immune regulation and immunotherapy.
The main advantages of this technique are that mapped peptides are known to stimulate Qa-1-restricted CD8 T cells, and the procedure is easy to perform.
The implications of this technique extend towards therapeutic designs for immune-mediated diseases such as multiple sclerosis.
Though this method can provide insight into Qa-1-mediated immune regulation in animals, it can also be applied to HLA-E-mediated immune regulation in humans.
To begin the procedure, design a library of 15 mer peptides in which adjacent peptides overlap by 11 amino acids. Obtain and reconstitute the peptides under sterile conditions. For each peptide, combine 5 mg of the peptide with 100 microliters of dimethyl sulfoxide. Combine 10 microliters of each peptide stock in a clean 1 ml cryotube to prepare the OLP pool stock. Then, load 20 microliters of each peptide stock into a V-bottom 96-well plate, and dilute each stock with 80 microliters of sterile water. Store both the remaining 50 mg per milliliter peptide stocks and the plate of 10 mg per milliliter peptide stocks at -20 degrees Celsius. Next, add 10 microliters of the OLP pool stock solution to a 15 milliliter tube containing 2 milliliters of irradiated dendritic cells in serum-free medium, thus diluting